Murray?
Yes, dear.
I have a question.
Which question?
I'm not really sure about how the DNA process works.
Which part?
All of it?
Okay. Let's review these so hopefully you'll have a better understanding of it. DNA replication has two stages, initiation and elongation. Initiation begins when the initiator protein binds to the origin of replication. This initiates the unwinding of the DNA. Helicase is the enzyme that unwinds the DNA molecule. Single-stranded binding proteins bind to each DNA strand to keep the DNA from joining together. The unwinding of the DNA molecule causes stress on the end of the DNA molecule. DNA gyrase comes in and relieves stress by allowing free rotation. The separation of the helix creates a replication fork. In order to synthesize a new DNA chain, a RNA primer would be added to the template DNA strand. The enzyme primase adds a free OH group for the DNA polymerase to work with. Elongation is the adding of nucleotides that are complementary to the template strand to create a newly synthesized strand of DNA. During elongation, DNA polymerase adds nucleotides to the 3' hydroxyl group. Therefore, elongation occurs in the 5' to 3' direction. In the direction going towards the origin of replication, DNA polymerase will add nucleotide strands continuously to create a leading strand. In the direction away from the replication fork, DNA polymerase will add strands discontinuously, and these would be called the lagging strands. The short DNA fragments on the lagging strand are referred to as the Okazaki fragments. The Okazaki fragments are joined together by DNA ligase. DNA replication is now complete. Do you understand DNA replication now?
Yes, I do.
Okay, now on to DNA transcription. Transcription is the first step in gene expression. During this process, DNA will be copied into RNA using RNA polymerase. During elongation, RNA polymerase moves in the 3' to 5' direction on the 5' to 3' DNA template strand. RNA polymerase unwinds the DNA strand, which causes a transcription bubble to form. Within the transcription bubble, RNA polymerase adds nucleotides complementary to the DNA strand. However, instead of thymine, RNA uses uracil to bind to adenine. This is the end of elongation. During polyadenylation, poly-A finds a signal AAUAAA, which marks the end of the important transcript. A ribonuclease then cleaves the primary transcript at the signal and creates a new 3' end. And poly-A polymerase adds a poly-A tail to the 3' end. After RNA splicing, this is our new pre-RNA. Lastly, RNA editing occurs in the pre-RNA, which allows the nucleotides to be changed, added, or deleted. To mediate translation, transfer RNAs are used. Translation occurs on ribosomes that coordinate the movement of tRNAs carrying different amino acids. A ribosome is consisted of two units, a large subunit and a small subunit. The large subunit has 3 compartments, which the tRNA moves through. When the mRNA is finished coding for proteins, a stop codon will enter the A site without an amino acid, and the mRNA and the polypeptide, as well as the ribosome subunits, will all be released. And this is the end of translation. We have now covered the central dogma of DNA replication, DNA transcription, and translation.
Thank you, Professor Murray. I think I can finally get an A on this test.
That is great. I'm so happy to hear that.